Cell suspension culture. As I told you that for the in vitro propagation of plant cells, culture medium is required. In biological sense, culture medium, it is any substance or gelling agent or liquid that provide essential nutrients for the growth of living organism. In case of plants, a combination of macro and micronutrients are required. In addition to macronutrients or trace elements, source of vitamins like thiamine and myo-inositol has also been supplemented in the basal formulation. In addition to that, a few amino acids, they have also been incorporated into the basal medium so that they can provide the organic source of nitrogen to plants. And most importantly, another component that is important, it is the carbon source. And here, the carbon source is in the form of sucrose. So all these materials, mainly they are formulated and solidified with the help of a solidifying agent, which is called as agar. So that agar, it will provide a solid surface where plant roots that are growing in vitro, they can be, you can say that they can establish there, like the soil. But if we want to grow cell suspension culture, for this purpose, we need a liquid medium without any solidifying agent. And cell suspension cultures, again, they can be formed from any cell type. Like कि कोई plant tissue से हम cell suspension culture बना सकते हैं, although कि ये थोड़ा सा difficult होगा कि अगर हम लीफ का पार्ट लेते हैं या स्टेम का पार्ट लेते हैं तो उसको लिक्विड मीडियम में एजिटेट करेंगे, कुछ सेल्स सेपरेट होके प्रोलिफरेशन तो स्टार्ट कर देंगे, लेकिन ये थोड़ा सा डिफिकल्ट प्रोसेस है. तो जो ज़्यादा इजी जो मेथड है वो ये है कि हम कैलस को यूज़ करें, विच इज़ अनऑर्गेनाइज्ड और अनडिफरेंशिएटेड मास ऑफ सेल्स दैट आई हैव जस्ट शोन यू इन द प्रीवियस पिक्चर. And it can be placed in sterile liquid medium, and then it can be agitated on the shaker properly, and aeration it is also managed so that depending upon the type of callus that from which plant it has been derived, then it may be very compact and sometimes it is very fragile and disintegrated very easily when it is shaken. So, if it is highly fragile, disintegrated easily, so in this case, the calli, they may be called as the friable. So, on the shaker, the cells, they may be disintegrated into a single cell or small cluster of cells or even larger aggregates of the cells, they can also be produced. But whenever they are growing in the liquid culture, then we also manage a proper combination of phytohormones like cytokinins or auxins so that proper cell division at optimum concentration of those phytohormones it is there. So single cell, if they are present in the liquid culture medium, then they will start multiplication. So single cell, it is dividing into two cell, and mainly they will develop ultimately in the form of aggregate because the daughter cells, they tend to remain together. Then with the passage of time, when cell division it will goes on, then nutrient depletion is there, and after particular time period, we need to subculture those cell suspensions so that they can be maintained for indefinite period of time. But one of the disadvantage of cell suspension culture are that some type of variations that are called as soma-clonal variations. They can be introduced so that their ploidy number or chromosome number may be changed at the somatic level. That can influence the cell vitality or plant vitality or vigor. So such type of disadvantages are there, but some scientists also notice that soma-clonal variations, for some purpose, they are also beneficial, but some sort of chromosomal rearrangements are there. But subculturing is required because, like bacterial cultures, cell cultures, they can also be entered into stationary phase cultures where there is no net increase in plant cell number due to the lack of nutrients. As far as cell suspension cultures are there, we can say that the cells in the suspension, in the presence of proper nutrients, they are actively dividing. They are more dependent on an appropriate level of phytohormones. Their ploid number, it can be changed, and their generation time or doubling time, the time taken by the cells to become double, it may be around 24 to 72 hours. And another characteristic feature of the cell suspension culture is that starch granules may appear in the cell. Nuclei, they become more distinct, and cytosol, it become more prominent or dense. So these are a few characteristic features of the cell suspensions cells. In addition to that, cell suspension cultures, they are having several advantages because cell suspensions cultures, they can produce variety of metabolites that are economically beneficial, and all those metabolites, they are secondary metabolites. प्राइमरी मेटाबोलाइट्स वो होते हैं जो कि सेल की ग्रोथ के लिए जो है वो रिक्वायर्ड होते हैं इन द फॉर्म ऑफ अमीनो एसिड्स या न्यूक्लिक एसिड, बट ऑन द अदर हैंड secondary metabolites, they are economically valuable in the form of certain chemotherapeutic drugs or proteins or enzymes.